Okay. Good Friday morning, everyone. Backyard Naturalists. Welcome to the Backyard Naturalist, hosted by the Urban Ecology Center, whose primary mission is to connect people in cities to nature and to each other. My name is Tim. I am your host this week. Uh, for the next half hour or so, I'm kind of asking everybody to put on pause everything that you may think you understand about nature and maybe life. I know that's a hard sell at the beginning of the saying this is gonna blow your mind. That seems like a pretty tall and maybe arrogant request, but I personally had my mind blown by this organism. You may be underwhelmed, but I don't think that'll happen. Um, you're here, and that says something because if I would have said today's program was about like kittens or river otters, I'm guessing we probably would have had some more people. But the fact that you're here with us, live or recorded, means that the right people are here now. So sit back and relax from the comfort of your screen on this cold, soon to be rainy January morning, at least here in Milwaukee, as we bring you episode 22 of season 5 of The Backyard Naturalist, Once Upon a Slime Mold. And a huge shout out to Amanda and James. So, Amanda for beginning to teach me the ways of Google Sheet editing. So, I'm a little pretty proud of this one. Um, and maybe stalling a little bit so we can all appreciate the beauty of my work here on this opening slide. Okay. Said it already. I'll say it again. Thank you for joining us, supporting the Backyard Naturalist with a nod to our local PBS stations. And I have two screens now. This is confusing me. All right. There we go. Thank you to the slimy and moldy people that make up the Backyard Naturalist subscribers. You are my favorites. I'm so lucky to be able to spend time with you and your thoughtful and silly imaginations. If you are not a subscriber, I encourage you to at least look into the benefits of a seasonal or annual subscription that keeps this program sustainable, keeps all the wonderful people, the wonderful guest lectures coming. And if supporting a subscription is not in your means, thank you for being part of this community. Um, this will probably be the last time I put this plug up, but we still have two openings on the UEC's Great Journey to the Backyards of the Galapagos in April, a once in a lifetime opportunity to visit a unique and magical world that is pretty much due south of Milwaukee at the equator. And if you're interested in traveling to the Galapagos with the UEC, let me know right away. Uh, One thing that I like to do here in Backyard Naturalist is kind of just keep a pulse on what's happening above our backyards. Our backyards are connected to the skies. There are a few things to report. So a great resource is the New York Times has an astronomy calendar. I love it. That kind of keeps you posted on what's going on. So this photo was taken just a few weeks ago. This is Jupiter's moon, Io, and this is taken from the Juno spacecraft just a few weeks ago on December 30th. So Juno has been dancing with and studying Jupiter for more than 7 years now. This picture was taken when the satellite was made a pass just like less than 1,000 miles from the surface of this, which is the most volcanic world in our entire solar system. Huge volcanoes, huge lava flows. And 1 week from tomorrow, Juno is going to make another very close pass and collect a lot more data and a lot more wonderful pictures that can help us better understand not only this moon, but Jupiter and the entire solar system and the universe, which is something that slime molds do also. If you have a chance, NASA has a really nice little photo album of this dynamic moon, and hopefully we'll be able to see a lot more soon. Then mark your calendar for February 15th. Not just because it's the day before Ethan Bott makes a triumphant return to host a Backyard Naturalist episode, but because of two expected events in space exploration. So the first one, Intuitive Machines, which is a company based in Houston, is hoping to launch its IM-1 mission, Intuitive Machines 1, which uses its Nova-C lunar lander, which is what we see here, and it's going to carry payloads to the lunar South Pole region. So again, there's a lot of companies that are anticipating all of the time that we're going to spend on the moon. And so part of that is setting up this little Amazon surface of deliveries. And so we're trying to get a lot of the stuff there before the humans come back. So there'll be all these stuff waiting for them when they get there. Um, this would make it just the second successful landing in the southern part of the moon. After India's space agency accomplished this last year, they were the first. And if this is successful, it will be the first ever lunar landing by a private company. So Intuitive Machines is one of 3 private companies that NASA hired back in 2019 to deliver payloads to the moon, along with a company out of Pittsburgh, Astrobiotic Technology, and one out of Edison, New Jersey, Orbit Beyond. But Astrobiotic was the first to launch in January, but it spectacularly failed. And Orbit Beyond is still a couple years away. But whoever is successful, this will be the first, in addition to the first private landing, it'll be the first American spacecraft to land on the moon since 1972, the year before I was born, with Apollo 17. So we'll see what happens on February 15th, see if this will finally be the one. And then also, there's a big development. The Japanese Space Agency is going to try to launch its flagship H3, which is built by Mitsubishi Heavy Industries. And it's going to be carrying a test payload and a pair of smaller satellites that, again, is really going to help us image the universe. So it's always fun for me anyway to track these events. But we are here primarily today to hear stories about a very unique group of species known as slime molds. So if you see something like this in your yard, you know, you probably will be overcome by amazement at the beauty of what looks like somebody skimmed the top off of a failed lemon meringue pie and threw it out their window, or had a failed experiment with peeps and just threw them out the window and they kind of plopped down on your yard. This particular species of slime mold is known as Fuligoseptica and has the quite distinguished common name of dog vomit slime mold. So dog vomit slime mold could be in your backyard. What is it? What is it? What is a slime mold? And I'll start by saying it's not a lot of things. It's not an animal. It's not a plant. Probably 9 out of 10 people would look at this, including myself, and say, oh, it's a mushroom. It's some kind of fungus. Can I eat it? It's not any of those things. Yet, it's a living organism. It's a living organism that moves around your yard searching for food right now. They move, they eat. Perfect fodder, and probably have been fodder for horror movies, just on a very small and very slow scale. So it's really hard to begin to even think about what this is, because for a lot of organisms that we feature here, we have like a good starting point. Like mussels, we had mussels a couple weeks ago. Those are mollusks. That's a good starting point. Vanilla is a type of orchid. Cockroaches are insects. So, but slime molds, there is no starting point that just makes sense to us that we can start with, that and move from there. And it's the same with scientists. Even though there has been a lot of increased interest in studying this for reasons we'll see today, we still don't know what it is. Scientists have moved the slime molds around the taxonomic orders. They did think they were mushrooms, which makes sense. So we kind of have to start from the beginning, or the most basic division of what life is. So at the cellular level, the the most basic level, organisms are grouped into whether their cells are prokaryotic or eukaryotic. So prokaryotes are thought to be the most primitive, the most basic blueprint for life at the cellular level. They are a cell, but they don't have membrane-bound organelles inside like a nucleus. It's just the stuff is everywhere. And it's a very large and diverse group, and primarily consisting of bacteria and archaea, which we have covered here in the past. We had the whey bacteria and Raiders of the Lost Archaea. And then eukaryotes are everything else. So single multicellular organisms, and the difference is that in eukaryotes, the cells have membrane-bound organelles, membrane-bound nuclei, and that's most of the life that we are familiar with. Again, the big three: plants, animals, fungi. Those are eukaryotic organisms. Slime molds are also eukaryotic organisms. Um, so starting out fairly straightforward, but again, they're not plants, they're not animals, they're not mushrooms, fungi. Uh, they're they're actually what we call slime molds, and what slime molds really are. There's a disconnect too, and that's a cultural thing. So we will put several different groups of organisms, several different phyla, clades, supergroups that aren't related to each other, and we'll call them slime molds. But there is a biological basis that is that helps us start off to classify what this group of organisms uh, are. Um, so we're not gonna like go down the that Russian doll nesting phylum, class, order, etc., which again is becoming less and less relevant anyway. But it is a good starting point to say that most slime molds are amoebas. They're in a phylum called the Amoebozoa, which is now my new favorite word. Um, and again, this classification system has been dynamic. It's been changing. Um, the Amoebozoa have been grouped with the protists. They've also been grouped with the protozoa. Uh, it's about as clear as mud, but protists are single cell plant-like organisms that aren't quite plants. Protozoa are single-cell animal-like organisms that aren't quite animals. So we're calling these amoeboid protists. You can also call them amoebas. You can call them protists. It makes sense. And amoebas are fun. Amoebas are more of a body plan than a classification, and it just refers to that blob-like way that they're shapeshifters. They can alter their shape. They can expand and contract. They're not limited to a cell wall. They can send out those little pseudopods. Um, and most cells can't do this. So that's kind of the first really cool fact about slime molds is they're just these shapeshifters um, at the cellular level and at the body level. Um, but with all these inconsistencies, again in the taxonomy, we're not going to go down that list, but we can start to describe what slime molds are. So we look at this beautiful, delicious cherry-looking set of fruiting bodies of a particular species, Cometrica nigra, widespread, usually found in forests and decaying woods. Um, and most slime molds are microscopic, but there's a huge group of them that are visible to the naked eye, and those are the ones that are where we will put in the field guides. Um, the life cycle involves a free living organism, um, usually single cell, moving around and then reproduce through spores. So similar to how mushrooms or ferns reproduce. Mostly terrestrial. There's a few that are aquatic, semi-aquatic. Some are parasitic, living entirely inside of plant hosts. And they're found all over the world, even in the driest deserts in the coldest Arctic communities. So um, we can kind of track our relationship with them and how we think of them. So the very first account of slime molds that is written, you know, humans have been aware of them for way longer than that, but the first written account goes back about 350 years ago. So the year 1654 for a species that we know today, or we call today, wolf's milk slime mold. At the time, it was called fungus cytocrescentes, or fast-growing fungus. So we pretty much always group the slime molds with the fungi, and it's probably easy to see why. And a kind of cultural result of that is that most of what we learned about slime molds have come from mycologists. Um, Eventually, the slime molds were taken out of the mushroom kingdom, but the mycologists out there are stubborn and they're like, okay, you can take them away from us, but we're still going to be the experts on them. Kind of like, this is our baby. We're still going to study them, even though they're not technically mushrooms. Yes, we admit. Um, so So it's still considered mycology, even though they're not at all related to mushrooms. So then the first thing after taking them out of the mushrooms is they're grouped into two major and useful classifications. So you have, you don't have to, don't try to read this. So you have the two groups, and this is what I remember learning in biology is you have the myxomycetes, those are the plasmodial slime molds, and then you have the Acraceae, which are the cellular slime molds. And this really allows us to understand uh, the two groups pretty well. Um, so it's reported that there's somewhere between 1,000 to 1,500 estimated species, like 700 or 800 described species. I gotta believe the actual number is much higher for a group of organisms that's almost as old as life itself. Um, we believe that slime molds go back more than 1 billion years. And so we're likely one of the very first types of living things. Um, but they come in all sorts of shapes and colors, all sorts of aggregates, colonies, honeycomb shaped, single cells. Um, and so when if we look at these two major groups, though, you see how different they are. Um, so um, we'll start with what are called the true slime molds, or the plasmodial slime molds. So these are again what we learned as myxomycetes, um, now referred to as myxogastria. Um, and so these are the ones that you'll see. These are the ones that you'll see in your backyard. They can get big. They can get like even in your backyard. Um, yeah, they can get like dinner plate size, 12 inches in diameter, um, which maybe not, that doesn't seem huge. That, it's a big chunk of slime, but it seems enormous when you realize that this is one cell, one single cell, a cell that you can easily see, no microscope needed. There's no divisions in there. There's no cell walls. This is one large communal organism, with thousands and thousands of nuclei in there. So it it moves and eats and reproduces with the stalks that release the spores, but it's one gigantic cell, just a huge party of cytoplasm. Um, and there is another cell that's kind of cool that we're familiar with in our bodies. So our muscle cells, our skeletal muscle cells, are also large. Um, one cell can be about an inch long and have lots of nuclei in it too. So it's not the only there and there's several other organisms that are single cellular that you can see with the naked eye. But to look at this and say this is one cell kind of blows my mind a little bit. And there's a particular species called the tapioca slime mold, which is what we see here. Um, the tapioca slime mold is worldwide, and it can reach like mammoth proportions. It can be like a square meter and weigh more than 40 pounds. So one single cell that weighs 40 pounds. And they can grow fairly big fairly quickly. Um, they eat, but they also just kind of join up with other free living cells. So you have two free living cells, they join up. Hey, let's share all of our stuff like a commune, and then they just become one giant cell. They merge, and so you have all these cells coming together to basically become one giant cell. Um, if you want to start looking for slime molds, which I really want to now, uh, you can usually find them, if you're going out to look for mushrooms anyway, if you have a period of excessive rain like now, but wintertime isn't the best. They're not a huge fan of cold, although they can be, some can be. Um, or if you've watered your garden kind of excessively, or you leave the sprinkler on or something like that, that's when they'll start to show themselves. So if you're looking for them, look for them, the rainier, the wetter, the hotter, the better. Um, this is also the group, this large group is what gave slime molds their name, because for at least part of their life cycle, they are indeed slimy. And that slime is important. In addition to being potentially massive, these single cell plasmodium slime molds, like I said, they move around. They move around usually because they're looking for food, um, or they're running away from something that they don't like. Um, but this movement involves just a fascinating uh, mechanism. So both within the cell and like shooting, being able to shoot out to expand these little fingers, um, they they kind of do an with like an exploration. And if they find something they like, they move towards it. If they find something they don't like, they move away from it. Um, if they've come across an area that they already have explored and they know that because they've left a trail of slime, just like a snail, they'll say, "Oh, I've already been there. I don't need to do there." So it kind of allows for spatial memory. Um, then they'll take part of it back into the main body and towards new areas that they haven't explored. All this happens very, very slowly, but so it's time lapse photography is our friend here when studying them. And it's okay that they move slowly since their main source of food is detritivores that also move very, very slowly. So it's like a slow speed chase between a slime mold and their prey. And this is their prey. Most of their prey is microscopic, not all of it. So they will feed on like vegetation. They are decomposers. They will also feed on the decomposer community. So they do love a good bacteria, good mold, good yeast. Um, they are detritivore specialists. They will eat other kind of microorganisms too. And then they eat like a good amoeba. They eat through phagotrophy, which is basically they engulf whatever it is their food and then incorporate that into that big party of cytoplasm. Um, so this is one of the main reasons they're different from mushrooms. So mushrooms will extend those hyphae and break down the food first and then use it to absorb the nutrients, whereas the slime molds just gulp it up like Pac-Man. Um, so sometimes predator, sometimes prey. There are plenty of animals that like to eat slime molds. There's a whole group of beetles that are slime mold specialists, both in the larval form and the adult form. And uh, slime molds have not made it past the gastronomic interests of humans without interest either. So uh, there are places in the world where slime molds are a specialty, including the dog vomit slime mold. Uh, it's grown in certain places. There's some, it's a delicacy in some parts of Mexico. There's a dish called caca de luna, which translates to poop of the moon, that people like to eat. So, if you look at all of the different slime molds, they come in all, like I said, all shapes and sizes, all colors. The only color that they don't really come in is true green because they're not plants. They don't have chlorophyll. So most commonly it's yellow and orange. Um, Less commonly are the darker colors, black, blue, green. And so here's a few. This is stemonitis. This is the fruiting bodies. So this one uses airborne dispersal. Not all of them. They use different types of dispersal, just like a lot of plants. Here's another good looking myxomycete. These are the fruiting bodies of different species, Diachea leucopodia. Uh, this is another photo of dog vomit slime mold, and its beauty. This one is called Trichiavaria. It is fun to look at all of the common names, but a lot of them don't have common names. Unfortunately, they're hard to find. So, uh, you got the dog vomit, you got the bird poop. This is called the false puffball, and I think a lot of us are familiar with the mushroom puffballs, the giant puffballs. This looks very similar. This is not edible like the giant puffball. Thankfully, a lot of and this is not a, don't take my word for this. Don't ever listen to me for this type of advice. But for the most part, slime molds aren't going to kill you like mushrooms can. Definitely don't eat it anyway. And most people think it's not worth eating anyway. None of them. But there are a few that you could consider them edible, but maybe you just got to put them in something that tastes good. Um, but again, for this one, in some parts of the world, they will fry them up, and probably a lot of butter, a lot of salt. And the spores of this one will can disperse through the air, through the water, and also through slime mold fly specialists that'll disperse the spores. Um, this is Metriticchia vesperium, and it uses a mechanical way to shoot its spores. They're ejecting, like the seeds of the witch hazel, or the touch me nots. They're ejected through a physical process. We met dog vomit slime mold. This is dog sick slime mold, a different species. Um, and this is a photo, so you can't see it. It's not all Harry Potter. But what this is doing, the specimen is in the middle of this movement. It's contracting that fractal network of the pseudopods. It's moving towards one direction, sucking in from another, looking for food. Um, and when it finds food, or if it finds food, it doesn't just stop there because it still can move. So it'll like nom 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 on this one, but it's also at the same time stretching out looking for more food. And if it finds two good pieces of food or three good pieces of food, it's going to make its body cover all of them and connect them with little tunnels where the nutrients can go. But that's a really important uh, process for some of the research that we'll learn about later. So they essentially want to connect to all the good food through a network of their own body. Again, one single cell taking a pretty interesting shape. So that's some of the myxomycetes, the plasmodial slime molds, the ones we can see. There is a whole entire different group of slime molds called the cellular slime molds. Equally fascinating. We just usually can't see them. And so the major difference with this group is that the cells don't grow themselves. They don't expand and become bigger cells like with the plasmodial. They stay a cell. They're microscopic. We cannot see them as individuals. But during some stages in their life, they become colonial. So they become a colonial group of cells working together. They become what's called pseudoplasmodial. You have your plasmodial and your pseudoplasmodial. They behave very similar to the plasmodial slime molds, but again, they're just congregating as tiny cells working together. Again, it can be to find food, it can be to eat, they can do it for defense, they can do it to move, they can do it to migrate, and then most importantly, they do it to produce spores for reproduction because they're um, too small by themselves, but together they can create structures like this. So they need to be colonial, and then when they're done with whatever purpose they've done, they'll break up again into the individual cells. So it's not like this big long party like the plasmodial. It's like a little convention. They get together, they hang out, and then they go about their separate ways again. So very similar to a swarm complex that we see in animals with a lot of kinds of animals like bees. Um, so this is probably the most commonly studied cellular slime mold forming a stalk in a fruiting body. It's Dictyostelium discoideum. Um, so in order for this to happen, and this is maybe the first time my brain just started to go, oh my God. So you have to have the cells coming together to migrate to this spot. But to make a stalk like this, some of the cells have to basically sacrifice their body. They go to the point, they die, and they become this kind of cellulose bundle of fibers. And that's it. Their life is over. And then more come and die on top of them. And then when the stock is sufficiently high, another group and the same species will come, and they're called slugs, and they'll just climb to the top and they'll become the spores. They'll do the spore, the reproductive process. So there's no genetic difference between them. They're independent, and yet they come together to make this way of reproduction. And so only the ones at top are going to reproduce to form the next generation. And they essentially, a lot of them in this type of form are clinging onto animals. So the animal comes through, takes the spores, drops them off somewhere else. So they do have to get above the ground somewhere. And but these are microscopic, so it's going to take a lot of them to make this stalk. And um, this isn't the only case where they exhibit altruism, if you're going to call it altruism. I know uh, altruism is a complex topic, and we need to have an entire uh, episode dedicated to it. But this is about as true altruism as you can get, sacrificing your body for the good of the colony. Um, they'll do it in other ways too, in both the plasmoidal and these. So if if a colony or an individual comes across a bacteria that becomes infectious and is at risk of killing the whole colony, the basically the part or the cells that have come in contact with that infectious part are going to detach from the group and contain it and die. So basically saving the rest of the colony from the infectious bacteria, essentially, like I said, sacrificing their own lives for the good of the colony. They're doing all this without a brain. They don't even have a simple brain. They don't even have any kind of neural network at all. Um, so again, for both kinds, the cellular or the plasmoidal, they can move towards, they can move away, they can move away from danger. Um, the the cellular slime molds move with these little pseudopods, like an amoeba. Um, the plasmoidal slime molds move by expanding and contracting their cytoplasm. A lot of it forms these regular geometrical patterns, these fractals. It happens slow. The fastest slime molds are clocked at about 1.5 inches an hour, and those are the fast ones. You can't see them move unless you're very patient or you have a camera, but we've learned a lot about them by tracking their movements with time-lapse cameras. Um, at the end, we'll take a look at some videos. But getting back to this idea of movement in an animal without a brain, um, what we've learned is this is the basic for what we're learning. This is the meat of what we're learning about slime molds, is this pattern of exploration. So let's meet. This is Physorium polycephalum, and this is referred to as the blob. And so this is a lot of the research has been done on what we're calling the blob, considered by some to be the most complex intelligent slime mold. It's plasmodial, so this is all one single cell. Uh, and like again, what we're probably seeing here is that there's two sides of this tree that have a lot of food. And so this one cell has spread out, it's eating on both sides, and it's connected by these little tunnels, these network of tubes. All the same organism, all the same cell branching out. And some of the best known experiments, the earliest experiments, came from a Japanese researcher, Toshiyuki Nakagaki. So the first thing he did is that he figured out that the most important thing is he figured out that slime molds really love oat flakes. So if you want to get the slime mold to do something, this is their Scooby snack. If you put a slime mold in a petri dish and you want to get it to move, you offer it these oat flakes. We still don't really know how they sense the food. It's likely through a chemical sensory system, but it drives them nuts, and it makes them want to eat them. So this seminal study here is that he created basically a fairly basic maze, and it's a simple experiment. Put a slime mold on an oat flake on one side of the maze, put another oat flake on the other side of the maze. So now you have two food sources. It's actually not the oat flake that they love, it's all the microorganisms that are on that oat flake that they really love. So the slime mold is happily eating away the other oat flake. It senses the other oat flake, so it starts to spread out looking for it. And when it does this, the first thing it does is it essentially fills the entire maze. It puts out that fractal network everywhere. Then it's either going to get a dead end or it's going to get to the food. Once it gets to the food, it's still going to be spreading out, but all those places that are dead ends, it's going to contract that arm. It's going to bring it back in and leave that slime to know that that's not a good place. So when it finally contracts and it gets to both pieces of food, what you're going to have left are the two blobs that are eating the food, and then you'll always have the shortest possible connection between the two in the maze. Every time it never fails to reduce itself to the most efficient connection, the shortest connection. Um, so on the upper left, you see the kind of exploring phase, and then eventually it gets to goes through the lower left and the upper right, and then eventually it's just the connection. So if there's just one path between the two, the slime mold really can only make one connection. But it gets really interesting when you offer two or three or four. It realizes there's many ways to the food, but it always reduces itself to the shortest network, and so uh, essentially this is where it becomes an algorithm. Um, this is where scientists say the intelligence begins in this animal. So um, no matter how complex that maze is, maybe there's 10 paths, it's still going to find the most efficient one. So now we move to research by at Hokkaido University. And so what we're seeing here in this first photo is that you little yellow, so this is in a petri dish. The little yellow blob is the slime mold. All the little white blobs represent little piles of ground oat flakes. The solid white line represents a border, so it can't pass beyond that border. So now we have a hungry slime mold in the middle, surrounded by all this yummy food. 5 hours later, you can see the slime mold is starting to spread its little hungry network of tubes to find the food. It's starting to find it. It's not fast, but it's hungry. 3 hours after that, the slime mold continues to spread out, finding more food. 3 hours later, continuing to spread out. And then here we are 16 hours later. And the slime mold has found almost all of the food. So it's essentially done with this exploration phase. It knows where the food is. Uh, and so now it doesn't want to just be spread out all over the place. It wants to concentrate its body on the food and the connections between the food. So it contracts all of those feelers. And then at the end of the experiment, you have one slime mold spread out to all these feeding stations, connected by all these little networks of tubes that are ferrying the nutrients around in the organisms. Again, no cell walls, no divisions. They're, I mean, kind of like blood vessels, but the nutrients are transferring in this one cell. What the slime mold didn't know is that the placement of these piles of food weren't random. They represent population centers of the greater Tokyo area. And then incredibly, this network that was created by the slime mold to basically connect all of these population centers matched up really well with the the actual subway system of Tokyo. So essentially, the slime mold in about a day found the most efficient way to connect all of these points. What took human engineers many, many, many years to figure out. Uh, and the other part of this is the researchers also were able to put in like some barriers that you couldn't just have in a petri dish. And the best way to provide a barrier for slime molds is light. They don't just they go away from light. So they also put into this model if there was like mountain passes that wouldn't make sense, um, or lakes or rivers or something, or maybe lakes, that it wouldn't make sense to put a subway through. And so they were the actual barriers. And so the slime mold stayed out of those areas, even though it might be more efficient. But it pretty much recreated this system. And so with this in mind, slime molds can be used to model other systems, or even in this system, they could be used to help scientists figure out alternative routes. So maybe there's a system outage, or maybe there's a crash that prevents one, it's kind of like a Google Maps, and the slime molds can then help create the next most efficient network out there. And it's all in its genes. So it can be a super useful tool in urban planning or even mathematical modeling. And it doesn't stop here, thankfully. So slime molds in a petri dish were successfully able to predict and help find lost Roman road networks simply by putting in the old Roman population centers, and it recreated some of the roads that we knew about, but it also helped archaeologists find lost roads they didn't know about because that was the most efficient way. So uh, they're becoming useful in archaeology, in urban planning. It's hard to understate that this is all happening without a brain. Not any kind of neural network. Even jellyfish have a neural network, but slime molds don't. So then it brings the question of, is this intelligence? Is this consciousness? And some scientists are saying these studies are beginning to challenge what it means to be intelligent and to be conscious. In the 1960s, physicist Evelyn Fox Keller started to really explore the idea of or really the question, can we use mathematics to model biological processes? And now that's a very common thing. But she met Lee Siegel, who was a mathematician from MIT, and then they started to seriously explore the mechanisms using slime molds as their pattern. And uh, not only the pattern that we're seeing in those experiments, but what is the pattern that allows the cellular slime molds? So what allows them to work together to build structures to move, to eat, and reproduce as one without any kind of neural network? Because again, these are individual cells working together, fully coordinating. And so the first thought was maybe this is like animal hive systems. Maybe this is like an ant colony. Maybe there is one cell in a colony that's acting like the queen and is sending out signals. They do know that the slime molds can signal each other with a protein similar to ATP. So the thought was maybe there's a queen, maybe there's a leader, maybe there's a pacemaker that controls the other cells and says, hey, we need to build this stalk. So you're going to become the stalk, you're going to become the spores. It's very similar to how ants work. They have a leader, and then they also have this algorithm of what to do. But that idea was pretty quickly dismissed. There is no one leader, and the slime molds really led to something we call decentralized decision making. So it's not a top-down system, it's a bottom-up system. There is no one leader, but anybody can be the leader. So yeah, we can use this for government classes too. So you picture a group of cells, they're all programmed, and their program is saying, let's look for food. Now I'm the cell that finds the food. I'm the cell that's closest to the food. I become the leader. I'm going to send out a signal. It's a cyclical AMP. I'm going to send out the signal, and I'm going to control the colony and say, hey, there's food over here. So whoever finds the food essentially becomes the leader, I don't know, the signal caller, and then the other cells follow the lead. So anyone can become the quarterback. Um, and then probably the best analogy in humans, or not humans, in animals, isn't the bees and the ants, but it's a flock of starlings. So you've seen the videos, starlings moving together in perfect coordination, like a dance, like a ballet. It's been, studied over and over again. It's beautiful. It's breathtaking. But this is also a case where there's not one bird that's in control. Every single bird in that murmuration has the power to change all of the other birds. They're all potential leaders. They all are leaders. Um, and so really what it comes down to, and I don't mean to simplify too much, but it comes down to that with the slime molds and the starlings, they're an algorithm that likely has very simple rules. So we've tried to model it. Keep an eye on three of your neighbors, do what they do. If, so that's your program. If you see a hawk and you're the first bird to see that hawk, you're going to move away. The other birds are going to follow you away from the danger. So anyone could have seen that hawk, but one person causes the starlings to move. If they're all on the ground, the first bird to notice a threat will fly. All the other birds will follow it into the air. We don't know what these rules are. We don't know exactly what they're studying, but if you think of a program, if A, do B, if B, do C, uh, it's probably a very simple set of rules, an algorithm that's allowing for a massive amount of beauty. Uh, and and this is where you get into artificial intelligence too. If we're creating artificial intelligence, we don't need to put all of the information into a machine. we need to give that machine a simple set of rules that will allow it to essentially think for itself. And the simplest programs allow for the most complex set of outcomes. And so this might be how we'd use nanotechnology. In the cockroach episode, we talked about how cockroaches are being studied like to be because they're so good at being in your house without you noticing them. They could also be the basis of a cleaning system that hides. And then, 2 o'clock in the morning, all these little nano robot cockroaches come out of their hiding places, clean up the crumbs, wash the windows. Um, and what they really need is a code. They need an algorithm. So I don't know how it would look. Maybe they're the tiny cockroach robots that wash your windows. So their code says, go towards a full spectrum light source until you come across smooth surface called glass, then start cleaning, leaving a trail, recording your steps, be tuned to where your neighbors are, a little more complex than a Roomba. Um, Communicate once you're done, go to the next window. Somehow your neighbors know that this window is clean, and then your job is done, your code is over, you go back to your hiding place. So that's the essence for a lot of this. It's a simple code. And if this isn't crazy enough, slime molds, these independent depositories of cellular stuff, have also been shown to have memory. So an experience will cause a memory that will cause their behavior to change, that they will remember, and they can pass on that knowledge to slime molds that didn't experience it. And so that's the basis of our next experiment. So this one, we're going back to the blob, the blob that engineered the subway networks. And so scientists set up a very simple system back to the reward system. They put the slime mold on one side. They put the food out, but this time the blob has to cross a bridge to get to the food. So there's only one way to the food. They have to cross a bridge. So the blob's like, okay, that's easy. I'm just going to go get it. But as soon as it touches the bridge, it 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 senses a deterrent because the bridge is coated in this salty solution. The salt causes um, the slime mold to be like, wait a second, I don't know if I can get to that food now because the salt, I'm not sure I want to touch it. I don't know, it might be dangerous, but I really want that food. So this is where that kind of sacrificing happens. They basically, one little tendril of that, um, the slime mold goes and engulfs the salt. And it's like, I'm either going to die or I'm not. Um, so it goes around it. It kind of goes dormant for a little bit, or it goes through a period of inactivity. Then essentially, it it what it normally would do is if it sensed the salt, it'd be like, here's a barrier, I'm going to go around it. In this experiment, there is no way around it. The only way through is through the salt. So then that little blob figures out, okay, I didn't die. The salt is okay. I still don't like it. Um, but it's not, it's not going to kill me. Uh, so essentially it kind of ate it, and then decided it was okay, woke up, said, hey, I'm okay. I don't like it, but I really want the food. So I'm going to cross the bridge. It's okay for us to cross the bridge and get the food. So not only did it have trial and error, um, it learned that the salt was not noxious, even though it thought it might be. And if in the future now, this slime mold comes across the substance, it will know that it's okay. It doesn't have to do that testing anymore. It can be like, oh, I can go across this bridge. I know I've seen the salt. I've experienced it. It's okay. We can go. Um, so a slime mold like this, again, it often grows when multiple organisms merge together. So scientists use that ability to basically divide an individual into smaller working parts. You can put them together. You can also divide them. So when they took a slime mold that learned that the salty coating was okay, then they kind of broke it up and refused it with parts from another slime mold that hadn't had that experience. And so they call them the naive slime molds. The experienced slime molds were able to pass that knowledge onto the naive slime molds that they merged with. Um, and then the new naive slime molds learned that the salty item is okay, even though it didn't go through that trial and error it was just that experience passed on. It's like, oh, okay, this is okay to cross. The ratio didn't matter. It didn't matter if you took a tiny piece of the experienced slime mold and put it in a huge bit of the naive, or vice versa. The ratio didn't matter. When the parts merged, they were able to share that knowledge through that, the pathway of networks. So I want to call them neural pathways, but again, they're not neurons. In fact, they're called non-neural organisms. So then if that's not enough, sometimes slime molds will, like a lot of good organisms in our backyard, our trees, everything, they'll enter a dormant phase, based on, the weather conditions, the environmental conditions, and they'll kind of temporarily shut themselves down, like a little hibernation, like, putting your computer to sleep. And then when they wake up from that dormant phase, they still retain the memory of that salt substance that they had when they went to sleep. So slime molds are exhibiting trial and error, learning, short, long-term learning, memory, passing on knowledge, essentially teaching other slime molds based on their experiences. And another tool that they have at their disposal is what I talked about earlier. After they visit a space, they leave that small um, snail-like trail. So then if they come back to that place, they either know that they were already there, or they can sense, that their neighbor Gerald had already been there, so they shouldn't go there because it's already been explored. And they retain that knowledge as spatial memory too. So add spatial memory to that list of indicators of intelligence. Slime molds were also able to learn and retain the memory that, in addition to salt being a substance I don't like, but I'll be okay with, they also were able to learn that coffee, even though initially it's like, oh, what's this? It's coffee. They find out, they taste it, go to sleep, come back, they realize it's okay. And then they realize, okay, coffee is also okay. Things like quinine, they're okay. So they're able to learn about individual substances being okay to them or being harmful to them, by absorbing them, seeing what happens, and then remembering what happens. So, and the story doesn't end here. So, uh, it's really become a hot topic. Now scientists are trying to not only really trying to figure out this algorithm that slime molds use in their daily lives. They're also trying to reverse engineer it. So we know that the pattern of exploration appears to be fractal, and we see fractal patterns all over nature. So they're kind of spreading out in all directions based on, this very probably simple algorithm. If I encounter this, I do this. If something good happens, I create this network of connections. And we're trying to figure out this code and use it for something much greater, much, much greater, which is now mapping the universe. We know so little about the structure of the universe. Um, this is the visible portion, the universe, the galaxies, the stars, the planets, these massive clouds of gas and dust. The things that we see are about 5%, constitute about 5% of the matter of the universe. About 25% of the matter in the universe is something we call dark matter. We can't see it. In fact, dark is a terrible term for it because light actually can pass through it. It doesn't even interact with light. We can see through it. We just can't see it. We have no idea what it's made of, but we can detect it, and we know that it's probably there because of its gravitational component. And it's likely involved with essentially holding the universe together with the network. Sound familiar? And it's kind of silly that I'm showing you a slide about dark matter because we can't see it and we don't know what it looks like. But the rest of the stuff in the universe, so that's only 30%, the visible and the dark matter. 70% of the universe is something we call dark energy, and that plays a role in accelerating the expansion of the universe, kind of tearing it apart. So dark matter and dark energy are really the things that determine the structure of the universe. We can't see them. We don't understand them. But scientists are now using the models of slime molds to try and map the structure of dark matter and dark energy. So again, modeling can be extremely useful in helping our brains predict and imagine and test. And this might be a very useful tool in our quest to understand the structure of the universe that we can't see. And so scientists are creating 3D maps of the known universe, the stuff we can see them, putting them in a model, and then releasing the slime mold code to predict where all of the dark matter is that holds the universe together, and where it lies. And it's kind, I mean, it seems like a huge leap, but if you think about it, it's actually not that. I mean, in my mind, it's not much of a leap to think that the same set of principles that allows that slime mold to connect food could be the same or very similar set of principles that connects the galaxies in the most efficient way possible if it's using a very basic formula. All this coming from a slime mold. And I do think it's very important to incorporate the research from our fellow backyard naturalists. And Jenny brought to my attention two very important references. So first, she found a researcher, Leslie Garcia from Mexico, who designed a machine that can translate electrical energy from organisms into sound. Um, and obviously there's a little bit of artistic license in there. And then she also found a great song about slime molds being anti-capitalist with an anti-capitalist message. Awesome lyrics. I can't share these videos right now for copyright reasons, but I will share the links to them in my next weekly email, and we'll also look at a couple of them after this is over. Um, there are just too many great videos and stories out there. Uh, if you find yourself doom scrolling and do something a little more productive, just go down the rabbit hole of Googling slime mold news and research. There's a great TED talk, great intersection of biology and social sciences and art. This photo is a musician that took the technology of transposing the electrical signals of slime molds into music, and then he performed a duet, he on the piano, the slime mold on itself. And so what I'm really left with as we wrap up our stories about slime molds, and it's gone a little long, is that just as when we looked at artificial intelligence, the simpler you can make your algorithms, the more powerful that artificial intelligence becomes, because it's less work that you do in terms of input and more work that the AI can do faster and better than us. And so we look at a blob that looks like dog vomit or bird poop in our yard, and that organism is demonstrating that its success comes from that simplicity through the code that it's embedded in its DNA. And I think of we there was in school there was something called Science Olympiad that some of us participated in, and there's science events that you compete in. So design and build a bridge out of toothpicks that can withstand the most weight. One of them was make a helium balloon rise to the ceiling as slow as possible. Um, and so from this I can imagine an event that says, here's a maze. There's food on the other side. There's 20 different pathways to get to the food. Be the first team to code your robot to find the shortest path through the maze. It could be very challenging, and I think the team's ability to succeed would be proportional to the complexity of the maze. So I imagine if there's one path, maybe the humans do it before the slime molds. But if you have 20 paths, I'm going to put my money on the slime molds. You bring in the slime mold, you put it on one side of the maze, you have it connect the entrance to the exit, but then it's able to very quickly find out the most efficient way between the paths and of the multiple paths. And slime molds aren't the only organism that are challenging all of our assumptions about life and intelligence. There's evidence that supports that trees are much more complex than we thought, with hive-like skills where a community of trees, even non-related species, start to exhibit signs of intelligence and problem solving and possibly even sentience. People are studying the hyphal network of fungi that act very similar to our brains, exhibiting problem solving. And then I take the next step to think about our own brains, our own consciousness. And I mean, are we not also defined by this complex neural network that is a result of an algorithm, that is a result of a code? So yeah, it can get pretty heavy here, but humans have been thinking about this for as long as we've been around. Einstein was looking for this universal theory to explain the universe. We're trying to figure out a universal theory that combines Einstein's relativity with the quantum world. Spiritual leaders are often seeking the truth or becoming one with the universe. Our complex equations have taught us a lot, but maybe the answers lie in the opposite direction, because it seems like we need to look for the simplicity. If simplicity allows for a greater complexity, then it's easy for me to think that the set of principles that holds the universe together in the most efficient way is the same set of principles that allows a slime mold to feed in the most efficient way. Or maybe the slime molds were the alien forms that brought life to Earth. and maybe all life in the universe is governed by the same principle, which makes me feel much more connected to all life, including slime molds. Because in a way, the simplicity allows us to let go of those barriers that might be preventing us from learning the truth. Some asked if slime mold we would consider it God, or does it hold the clues to understand what we call God is? Or does slime mold hold the clues to help us better understand what life is? Life to think that life can be refined by an algorithm sounds maybe crappy, sounds awful. But if you open your mind to it, if I open my mind to it, it doesn't make life less magical to me. It kind of makes it more magical. I don't feel more alone. I feel more connected to all the living and non-living things, and suddenly things that we chalk up to like coincidence start seeming less random and more intentional. All this from a blob in our backyard that is always there, whether we see it or not. It won't hurt us. It won't hurt our pets. That really took a deep path at the end, but I just, thank you, Blob. Thank you, Blob, that I know is not a mushroom. Thank you, Slime Mold, for reminding me of the power that comes from simplicity, and for helping me challenge my assumptions of intelligence and consciousness, and for making the universe seem a bit smaller and more connected. Whew, okay. So that's it. I'm going to stop sharing my screen.